So, who are these micropollutants, or persistent organic pollutants, or xenobiotic organic pollutants? You can find all sorts of terms to describe these chemical substances. They are predominantly manufactured via industrial chemical processes, which I'm sure that you will discuss more at length in detail with my colleague in his bank. Um, One common thing about across these various chemicals is that they are absolutely alien to the environment, which means that there are no specific enzymes for their transformation or degradation. In the most abundant taxa present in biogeochemical processes. Oh, there are lots of things in this one line. So when you think about a chemical structure such as glucose, and a given glucose concentration, as we learned, would represent a chemical signal to microorganisms. That chemical signal would cascade through through the microbial metabolism, and that would induce the translation of genome sequences that would eventually lead to the expression of enzymes to degrade glucose. Now, that's exactly what we mean here. For these chemicals, there aren't any recipes, so to speak, in the genetic libraries of most of the microbial taxa. That doesn't mean that it doesn't exist, or parts of it wouldn't exist a good recipe for to express enzymes, but most of the microbes don't have those recipes, so to speak. And those kind of aspects are really interesting, because when you think about these chemicals, and we will take a look at the chemical structures, very complex, very complicated structures. Parts of those chemical structures might be recognizable to some of the microorganisms, or at least their metabolism would be expressing some enzymes that could partly cleave and transform these chemicals. So it's very convoluted, very complex what actually occurs to those chemicals, which has fueled an enormous body of research in the past 20, 30 years. The examples of xenobiotics and associated environmental systems we will focus our attention to here during this lecture are trace organic micropollutants present in municipal wastewater. How did they get there? So the most prominent chemicals as such, or xenobiotic micropollutants, are represented by pharmaceuticals and personal care products. Through human metabolism, they would be excreted primarily via urine, and would end up in the sewage and be transported to a treatment plant. So that's how it happens. Now, through human metabolism, we are also aware of the fact that some of these pharmaceuticals, the metabolism or pharmacokinetics is very complex. And very often these active substances in our medicines would be transformed by the human body, by the human metabolism, and thereby forming so-called metabolites or conjugated chemicals. And these chemicals would propagate through the system. Importantly, when it comes to wastewater treatment processes, and particularly the conventional types, they are not designed and or optimized to remove these chemicals. Since those designs have been developed, say, 50, 60 years ago, and by that time it was by no means possible to quantify these chemicals. It also wasn't picked up on the radar of engineers and regulators, and therefore our current design doesn't really is not optimized to remove them. Now let's take a closer look at these chemicals. So here I'm showing you a incomplete list of pharmaceuticals, and this list is also from Norway, where a large part of my research focused on micropollutants. And one of the first things that you would be looking at is statistics and is statistical data from authorities, and on that you could assemble a list of chemicals and the associated amount of chemicals that have been sold. And in Norway, in 2007 or 8, these amounts have been, of pharmaceuticals have been sold. You can see some of them are really huge numbers. Paracetamol, highest. You can see diclofenac, which is a very, very problematic chemical. Over a ton or almost 2 tons have been supplied. Ciprofloxacin, very problematic antibiotics. Tetracycline as well. You can see that these are large quantities. When you go down to the treatment plant and you take some samples and you start calculating how much of these chemicals actually appear at a treatment plant, you could see some hormones as well, so like estrogens would appear probably in the grams of mass loads per day at a treatment plant. You can see that the magnitude higher values would correspond to that are emitted in the form of some important antibiotics like ciprofloxacin, trimethoprim. It's in the 100 grams. Or, as we've seen it for ciprofloxacin, it actually would be 2 magnitudes, so it's around a kilogram, which is an enormous amount. In fact, this particular data has fueled quite a few studies later on after we published this data, and indeed they also found that very high amounts of ciprofloxacin, which is primarily down to the fact that this is also a chemical that is transformed from another pharmaceutical, and so the two are adding up in the stream. So very complex processes. The last 2 ones are also antibiotics, sulfamethoxazole and tetracycline. Here you can see average concentrations for these chemicals that I show here. So here, one of the things that you can see and appreciate here is their relatively low concentration. So I'm showing you values in nanograms per liter. So how does this go? So milligrams, thousandth of a milligrams per liter is micrograms per liter, and the thousandth of micrograms per liter is nanograms per liter. And so these are very low concentrations, and as I said, they haven't been picked up and this problem until the end of the 90s. In 1998, there was an important paper published on that. And that really, that was really possible because specific technology dawned on us that allowed us to quantify these chemicals at these levels. If you now, after the concentration, look at the occurrence patterns, that is also, it also seems that it's very relevant to micropollutants as well, and more importantly, pharmaceutical micropollutants and hormones. You can also see that they have a diurnal variation. So here I'm showing you a data that I obtained in a 3-day measurement campaign. With the yellow, I show the volume of wastewater arriving at the treatment plant. And with these charts, I'm showing you the 8-hour flow proportional mass load specific to the total load. So it's basically 45% of, what is this? Astron arrives in the morning hours, and then roughly around 35% later, and during the night it's 20%. You can also see something else. Here I'm showing you cyclofluxacin and trimethoprim. And the occurrence of those chemicals also show very similar patterns to hormones. Part of the reason, as you may think, is that these are excreted through urine and together with hormones. Now, an important observation was that antibiotics that were administered differently, so you can see here the administration pattern, they showed a very different occurrence pattern. They had a peak load that corresponded most of the days with the hydraulic peak, the yellow column. So as you could see, that the propagation of these chemicals and pollutants from the human body to the treatment plant really had the fingerprints of human metabolism, transport, and all that. All those impacts has propagated together, and basically what we could observe at the influence of the treatment plant was that administration, pharmaceutical administration of these chemicals was one of the factors that seemed to have influenced the most their occurrence patterns. So such conclusions can also be drawn. I should also mention that there are other types of xenobiotics in wastewater as well, and in other streams, landfill leachates, etc. And these xenobiotics can occur up to a milligram per liter level. And prominent examples are the polyaromatic hydrocarbons, or PAHs. So those chemicals would be produced and would leach out from various constituents of our environment, or domestic environment, etc., which would trigger these relatively high concentrations. Nevertheless, they are not micropollutants. They are called xenobiotic pollutants, xenobiotic bulk pollutants, and they are different from those that this lecture is about. So let's take a look at a little bit of context, at the context of biodegradation, biotransformation. So what is really happening, and how do xenobiotics relate to the various substrates? So as we learned, primary substrates, or readily biodegradable substrates, are essential for microbial growth and maintenance. They're involved in growth, decay, and hydrolysis of organic matter, and the concentration of biomass in wastewater treatment systems is proportional to the removal of these chemicals. What is important that when we look at the removal of micropollutants, we can also see that the degradation rate, or the transformation rate, of micropollutants is also proportional to the biomass concentration. So that goes to show or indicate that the enzymatic processes that are ongoing in the cell in response to chemical signals of bulk substrates in the system can somehow act on these micropollutants as well. Besides primary, if there are primary substrates, there must be secondary, right? You would say. And these so-called secondary substrates are, so most xenobiotics constitute as secondary substrates. So they are chemicals either in the concentration too low to support the active biomass needed for the compound's degradation. So these secondary substrates, even if they are in a 1 milligram per liter concentration, that wouldn't be enough to fuel an effective growth. So that means that microbes would have to rely on bulk substrates that can fuel their growth, and these xenobiotics are by no means such substrates. So there is this reliance on primary substrates, and the secondary substrates are basically being transformed a little bit with enzymes, with accidental cleavage of these molecules by enzymes. And if they are being transformed or oxidized in one way or another, they could contribute to the electron donation process. So that's possible. However, their contribution compared to primary substrates is really negligible. Now, a subgroup of secondary substrates are chemotabolic substrates. And these substrates provide no energy benefits to the cells, so there is no accidental inference of electrons from the accidental oxidation of these chemicals. Again, the cell has an obligate need for a primary substrate, and importantly, chemotabolic substrates would also interfere with other metabolic processes, i.e. they would be toxic, and they could inhibit some parts of the metabolism. And as such, you probably noticed that I use the terms biodegradation and biotransformation so far. Why did I do that? So when we say biotransformation, what we mean is a partial conversion of the molecular structure. More on that in a little bit. So for micropollutants and trace organic, we can define a process rate of biotransformation. And that is basically a transformation rate that would occur in a typical unit operation, such as in an aerated bioreactor that fuels, for example, heterotrophic growth or autotrophic growth. So we would have these sort of bulk substrates present in our bioreactors, and given those substrates, given those enzymes expressed in a bioreactor, the resulting biotransformation rate of chemicals, i.e. this kind of accidental partial oxidation of these molecules, could be characterized with a rate. And that rate is denoted with the K-bio acronym that stands for biotransformation rate. And it depends on the biomass concentration and the dissolved micropollutant concentration. Here you may notice that I'm using a different acronym from S and X, denoting soluble and particulates. So sorry for the confusion, but somehow convention through international literature uses much more this kind of notation. So I will just use that. So this will have a specific kinetics which we will discuss in the next slides, that we call as pseudo first order kinetics, and the unit is litergram per day. So the unit for this parameter kbio. So what does this pseudo first order kinetics actually mean? It means that it is first order kinetics in regard to the substrate concentration, okay? And it is pseudo because you also multiply kbio times the CLI by the biomass concentration. So that's why it's sort of that's why it's referred to as pseudo first order kinetics. And that's basically a simple linear ordinary differential equation for the change in the concentration over time for these old micropollutant concentrations. As a homework, please take a look at this very simple ODE, or ordinary differential equation, and please derive the analytical solution. I integrate it, and for that consider excess constant as well as kbio. Okay. Okay, here's some explanation that I've already given you on the term pseudo. Now, finally, we can talk a little bit about, I can explain myself and the confusing use of, seemingly confusing use of the term biotransformation versus biodegradation. So when we talk about biodegradation, so it entails the complete mineralization of chemicals. So, for example, of readily biodegradable substrates into water, CO2 and water and some other chemicals. Now, when it comes to biotransformation, the process is limited. So you don't really mineralize, for example, sulfur methoxazole as a microbe, but what you're going to do is, well, it will depend on what kind of bacteria you're considering. So here you can see something like ammonia oxidizing bacteria converted into various forms. It would put an acetyl group or moiety on the chemical, or a nitrite group. Other microorganisms would do something else. But overall, what you can see is that it's by no means CO2 and water that is the end result, but it's basically just a transformed molecular structure. Yeah? Something is oxidized here, cleaved off there, something is added to that, and that's it. So that's the main difference between biotransformation and biodegradation. Now, when it comes to the treatment and really the removal of these chemicals, in order to enhance the removal, what the engineers and scientists are looking into is to transform these chemicals to a level that so that the chemicals, the products that are forming, have much less toxicity, much lower toxicity, and they are much less harmful, therefore, to the environment. After all, these chemicals are highly active, and this activity and the loss of this activity is what the main purpose of treatment is. Not really the mineralization. We don't want to go that far, or we can't unless we invest a lot of energy, but treatment's goal is to reduce or completely lose the activity of these chemicals. Now, let's formalize a little bit what happens to these chemicals in bioengineered systems. And for that, I drew a Pac-Man here, and I have the particulate matter, etc. And so let's try to simplify the system and create a model of this system. So on the one hand, we have the biotransformation using this Pac-Man, and that is the transformation rate is proportional to the concentration of dissolved and bioavailable micropollutants. Another part of the micropollutants will, so if it's not dissolved, then it's somehow associated, so sorbed, whether it's adsorbed or absorbed, we don't really know, but it's associated with particulate matter. Now, according to thermodynamic principles, the more of these chemicals we would remove from the aqueous phase, the more we would push desorption processes, i.e. the chemicals would desorb and get into the aqueous phase more from the particulate matter. So that means that in our system where the biocatalyst is a solid, a particulate matter, we will have these trace organics in two compartments of our system. So they will partly be sorbed and associated with particulate matter. The other part will be dissolved and bioavailable. And this equilibrium, there is an equilibrium between these two compartments, that thermodynamic equilibrium that can be just very simply characterized by a sorption coefficient, or as we denoted it, the KD. And the sorption coefficient can be calculated as a function of the concentration of trace organics associated with the particulate matter, the trace organics in the aqueous phase, and the biomass in our reactor, or the particulate matter, right? This is the particulate matter in our reactor. Moving on, some more process rates to include into our stoichiometric matrix related to sorption and desorption. As I mentioned, these are equilibrium processes, but in some cases, desorption might be not very fast, and as such, we would need to account for the change in the concentration of these chemicals over time. And to do that, we can write up two rate equations, two process rate equations for sorption and desorption. And these are pointing to opposite directions, or opposite ways, in a way. We'll see that in a little bit. So sorption would be, again, these are both first order kinetics or pseudo first order kinetic equations, and the rate constants for sorption, desorption, and the ratio thereof would yield a KD coefficient as well. So that is based on this principle, one can express the sorption coefficient as K desorption times KD. And here is just a K desorption times the concentration of the chemicals in the biomass phase. Now, the systems are not that simple, and in addition to dissolved concentrations and those that are sorbed to particulate matter, as I set out earlier, there are also micropollutants that are conjugated, that have a little moiety chemical group attached to them, an acetyl group, a glucuronate group, etc. Now that means partly microbes are attaching these, or particularly not the microbes, but in the human body, through human metabolism, these chemical moieties groups are attached to trace organics in order to increase the solubility thereof, and thereby facilitating the faster release of these pharmaceuticals from the human body. But that also means that they are excreted from the human body in this conjugated form, and they would also arrive in this form to our treatment plant in larger quantities. So if we now look at the more complete picture, you can see that dissolved bioavailable concentration will be influenced, besides sorption, also by the rate at which this moiety can be cleaved off from the trace organic pollutants, and which would effectively also mean that these micropollutants can be retransformed into the so-called parent chemical. So these would be the parent chemicals, and these are the not so parent, the daughter chemicals. Okay, so these are the conjugated form, and these are the deconjugated forms of parent compounds. Depends on how much terminology you are willing to learn in terms of chemistry and biochemistry. These are the jargons that used in pharmacokinetics. Now, this retransformable fraction is really interesting. As I mentioned, human metabolites form a large part of this. It's also a catch-all because it's not just human metabolites, but other commercially available chemicals can also form this fraction. As I said earlier, ciprofloxacin can also be formed by a simple biotransformation by a microorganism from a commercially available other pharmaceutical called enrofloxacin. And so that's an example of how that can also appear on your radar or analytical instrument. There is another part that is associated with the matrix. It's a very complex matrix, particularly when it comes to municipal wastewater. And so a part of these chemicals are sequestered in particulate matter or hydrolyzable matter, and basically through hydrolysis of say proteins or other polymers that it's entrapped in, or they are entrapped in, they can also become bioavailable, i.e. dissolved. And that sort of hydrolysis rate would also sort of liberate the parent chemical. So it's a catch-all term as well, or acronym or state variable. And here is the simple stoichiometric matrix just for these four processes. As you can see here, the process rate is there are numerous process rates, and those process rates are not related to microbial growth. And by now you probably also guess why. Because the transformation of these chemicals is not directly related to microbial growth, right? So the accidental cleavage of these chemicals and oxidation on these chemicals in the active enzyme sites does not contribute to a significant extent to microbial growth, and which is why it's decoupled from growth. Nevertheless, you can see in process rates that the biomass concentration is there for most of them, and through this it's not the growth rate that will influence the process rate associated with micropollutants, but it's the actual biomass concentration or biocatalyst concentration that will allow us to couple these models or fate process models with a model like a growth decay model. Okay. So here you can see the 3 state variables, and so for desorption, what happens through desorption is that the particulate concentration decreases and the soluble concentration will increase. So that's you can see with the -1 +1. Through sorption, chemicals would go from the soluble to the particulate phase. Biotransformation would reduce the concentration of micropollutants in the aqueous phase. And finally, the parent compound formation or retransformation would occur in a way that retransformable chemicals would be biotransformed back to the parent chemical. And indeed, there is a very large body of research that has been done in my group on this subject. So it's also nice to speak about it. You can see also the name of some of my PhD students from the past with whom I've done those work. So in order to quantify these parameters, one could do a almost a simple goal seeker one, maybe. Probably would need a little bit more sophisticated tools, but hey. So the K-bio you could derive by running an batch experiment. So it's a batch reactor that you would put some of the micropollutants into, and you would follow their concentration over time, typically a day. So we will be looking at this, how to do this kind of parameter estimations later on. K-bio and retransformation rate, or KDAC, can be estimated from batch experiments where we use a real wastewater. So in real wastewater you would have the parent chemicals, but also the metabolites that would retransform. So here you can see that retransformation, when at the start of the experiments, the retransformation was relatively fast compared to biotransformation. And once you retransformed all the conjugated metabolites, then in effect the curve would bend. So that means that you have a net biotransformation of chemicals. And finally, KD, the sorption coefficient, can be estimated also in batch experiments, where you would inactivate your biocatalyst, so you wouldn't allow it to transform it, and you would then measure the concentrations over a shorter period of time, and then estimate the KD according to the equation that I showed before. Here I'm showing you some values as well for these different parameters. There is also one that is very interesting here, and that's the last parameter, and that's the ratio. That parameter shows you the ratio of the parent chemicals to the conjugated chemicals in samples. And what you can also see here, I show you here some of the I show you the values, this ratio value, ratios for 3 important antibiotics. What you can see is that since the value is less than 1, that means that implies that the conjugated fraction is or the retransformable fraction is in a higher concentration than the parent chemical. Indeed, it can be twice as much, or for ciprofloxacin it's much more. And that's really interesting. We will discuss that more in semester 2. Here, let's suffice to say that when you quantify these chemicals in water samples, that can still, after 22 years, can be done using relatively expensive and fancy chromatography, mass spectrometry, or tandem mass spectrometry, which are pretty expensive instruments. It also needs a specific knowledge to operate, on and on. What it implies is that typically there exists a method to quantify the parent chemicals. Those are known to analytical chemists, and if not, then they develop a method for that. So that's fine. What about the conjugated or the retransformable chemicals? Well, retransformable chemicals can be present in very high quantities. Sometimes we don't even know what kind of metabolites, what kind of fractions they are being transformed to from. So that renders this fraction to be quantified only indirectly. So from observations that we can make, for example, on kinetic curves like this one. And in this using this experimental data, which is basically represent the dots or measurements of the parent chemical, the parameters that we would estimate are not only K-bio and K-DAC, but also the initial concentration of the retransformable chemicals, CCJ. And that's what you can see with the dashed line here. So this initial concentration is also quantified or estimated from these experiments, which would otherwise be very difficult, if not impossible, to quantify by an analytical chemist in other ways. So as you can see, it's an indirect measurement of or quantification of all these retransformable chemicals through these kinetic studies. And therefore, thereby resulting in these values, which are really interesting. Why? Because the analytical chemist can only see this fraction. And indeed, the fraction that they cannot see, cannot quantify, can actually be present twice as much or quantities or even more. We will discuss further the implications of this in semester 2. And with that, I thank you for your attention.